Good afternoon, ladies and gentlemen. Welcome. We are going to be discussing on-demand oral TDF FTC for PrEP. Is it an option? It's my great pleasure to hand over to my co-chair, who, if you were not here this morning to hear her brilliant speech, let me tell you, you missed something really good. Sheena McCormack, who is one of the leaders in our field, has been involved in HIV prevention, both in vaccines and in PrEP and microbicides for many years, and has led the way in many ways on trying to define new ways in which we can show that products are effective, as we saw in the PROUD study. So indeed, I'm very proud to have her as my co-chair and to hand over to her.
Thank you, Slim. Well, I've asked Slim to do the wrap up, and I'm hoping he's going to give us a wrap, because there's nobody who can wrap up better than Slim. We've got a great line of speakers today, and I think there's detail to discuss, and so we want you all to join in with the discussion as well. But in case you are not already aware, some of the guidelines have adopted the on-demand event-based dosing for gay men, so certainly in the UK we will do that. In France, the European AIDS Clinical Society recommend it, but it's not so clear in other guidelines. So I think this is a really important topic to have a discussion on today. Now, people are waving, but is that for a reason? No, just an accident. Okay. Okay, great. So now, did we say who's going to introduce? Oh, back to Slim.
And in case you're wondering about the air conditioning, I have already informed the technicians to try and get the air conditioning raised in this room, because I'm very aware you're cooking out there. As we are really cooking on the top here. So without any further ado, it's my great pleasure to start off by first thanking Jean-Michel Molina for conceiving this idea and the session, and for having the wisdom to put this issue firmly on the agenda. We are deeply thankful to him.
Oh, it's my pleasure to introduce Angela Kashuba. So Angela is a professor of the Eshelman School of Pharmacy at the University of North Carolina at Chapel Hill. She's chair of the Division of Pharmacotherapy and Experimental Therapeutics in the Eshelman School. And she's also the director of the UNC Center for AIDS Research, Clinical Pharmacology and Analytical Chemistry Core. That's a long line on your address, Angela. She's an adjunct professor of medicine in the Division of Infectious Diseases in UNC School of Medicine. Her lab work is focused on elucidating the mechanisms of drug distribution in tissues and predicting efficacy. She truly is the queen of pharmacology. Angela.
Thank you so much. I don't think I've ever been called the queen of pharmacology before. I kind of like it. So I'm very excited to be part of this session. I'd like to thank the organizers for their kind invitation. These are my disclosures. And for those of you who are in the session this morning where we were talking about topicals, don't think badly of me. I do have some of the same slides as in that one as I do in this one. But the principles do hold true, that the goals of pharmacology are to deliver the right drug to the right site at the right concentration for the right length of time. And these are the tenets that I'm going to be focusing on for this presentation. And obviously, we're not really going to be talking about the right drugs because we're talking about tenofovir FTC. So I'll be showing you data from tenofovir FTC to address these other issues, the biologic site, the concentration, and length of time. So the right sites that we're talking about are mucosal tissues, colorectal tissue, and tissues in the female genital tract, vaginal tissue, and cervical tissue. And though the epithelium might be different in these two areas, the cell populations are really similar. And so what we're talking about is where exactly should infection be aborted within the mucosal tissue. This is a cartoon of the early events in HIV infection, with HIV crossing the epithelial barrier, potentially getting taken up by dendritic cells, finding activated CD4 positive T cells to infect, with dissemination of virus and establishment of a lymphoid tissue viral reservoir. We think that the stroma is where we should really be targeting to abort HIV infection, although there's been some data to suggest that there may be trafficking of virus by dendritic cells to other sites. If we don't have high enough concentrations in the stroma to protect from infection here, then we certainly need it here in regional lymphoid tissue. Now, when we look at oral antiretroviral therapy, all antiretrovirals get into mucosal tissues to some degree, and this is a summary of those data. Pink boxes are female genital tract data, and blue boxes are colorectal tissue data. These drugs are binned by their therapeutic class, and they're also represented as ratios. The ratio of the tissue or the mucosal concentration to plasma. So anything that lies above that red line at 1 means that the drug is concentrating in the tissue. And from these data, you can see a few things. One is that there's significant intra-class variability, so not all drugs from one particular class penetrate a certain way into mucosal tissue. And we also see in general that concentrations in the female genital tract are lower than colorectal tissue. Well, let's just focus on tenofovir and FTC for a moment. And so you can see that these drugs are in these mucosal tissues at or above the plasma concentrations. And you can see tenofovir in colorectal tissue achieves very high concentrations. But I'm going to move away from tenofovir and FTC for a moment, because these drugs have to be taken up into cells and then metabolized to their active form, tenofovir diphosphate and FTC triphosphate. So we'll spend a little bit of time talking about those. And what I have plotted for you here are concentration time curves of these metabolites. On the left is tenofovir diphosphate, and on the right is FTC triphosphate. And these are data after a single dose of Truvada. And rectal tissue concentrations are in blue, and female genital tract tissue concentrations are in pink. And so you can see for tenofovir diphosphate, after a single dose of Truvada, the rectal concentrations are quite high, and they last for an extremely long period of time compared to concentrations in the female genital tract. So these concentrations are actually 2 logs higher, or the exposure is 2 logs higher than what we see in vaginal tissue or cervical tissue. Now, the opposite is true for FTC triphosphate, where those concentrations tend to be higher in the female genital tract than in colorectal tissue, but these half-lives are very short. By 72 hours, we don't detect this compound at all. So that's after a single dose. What happens after multiple doses? Sometimes the question is asked, well, how many doses does it take to get to steady state? Well, if we look in the lower female genital tract tissue, we actually don't see very much accumulation at all. These are trough concentration data modeled after the first 10 daily doses of Truvada, and it takes only about 2 doses to get to maximum accumulation of FTC triphosphate and 3 doses for tenofovir diphosphate. That's quite different than rectal tissue, where there is continued accumulation of tenofovir diphosphate and FTC triphosphate, so it takes about 6 to 9 doses to kind of max out that concentration. But when we're talking about tenofovir and FTC, we may be talking about more than just the active metabolites. These drugs compete for endogenous nucleotides to be incorporated into the elongating DNA chain of the virus. So, for example, tenofovir diphosphate is competing with DATP for incorporation. FTC triphosphate is competing with DCTP. And Gerardo Garcia Lerma, who was mentioned earlier, did a very interesting study where he saw as you increase DATP concentrations, you decrease tenofovir activity. So it's possible for these endogenous compounds to potentially outcompete our drugs of interest. So because of this information, we decided to measure DATP concentrations on the left and DCTP concentrations on the right in cervical tissue, vaginal tissue, and rectal tissue. And what we found, somewhat unexpectedly to us anyway, was that these endogenous nucleotides were found in lower concentrations in colorectal tissue, suggesting there was less endogenous compound there to compete for our drugs of interest and potentially have greater efficacy even at the same concentrations. So if we just look at the ratio of tenofovir diphosphate to its endogenous nucleotide, or FTC triphosphate to its endogenous nucleotide, we do see that there are some significant shifts in these PK curves, where colorectal tissue exposure is much higher than what we see in the female genital tract. And even though the female genital tract has higher FTC triphosphate concentrations early on, those ratios tend to be about the same. So how can we use these ratios now to decide how effective the compound is? Well, we decided to look in CD4 positive T cells and identify the ratios that are fully protective against infection in these cells. And so we did this for the ratio for tenofovir, we did this for the ratio for FTC here, and then we did some fancy mathematical surface modeling to understand the synergy when these two drugs are used in combination, and there clearly is synergy between the two. So with that, we were able to identify what the target ratios really should be for cellular protection and for mucosal protection against HIV infection. And so when we did this, we were able to ask the question, well, how many individuals would achieve effective concentrations at the end of whatever dosing interval they were taking? So if they were taking 1 dose a week, 2 doses a week, 3 doses a week, what proportion of people would actually achieve what we thought was a good target ratio, and we chose an EC95 for ours. We did this in colorectal tissue for emtricitabine in red, TDF in blue, and then Truvada in purple, and then in the female genital tract we did the same thing. And so what we found was that with Truvada in colorectal tissue, 2 doses a week resulted in all of the population that we simulated achieving this EC95 target ratio. And for female genital tract, it would take 7 doses a week, or once daily dosing, in order for the majority of the population to achieve this ratio. And our results are pretty similar to what we've seen for the MSM population and what we've seen in Partners PrEP. But the reason that we did this was not to try and mimic clinical studies. We did our analysis outside of any clinical study results. It was more to probe the system in terms of what drugs might be best used for each of these types of mucosal tissues, or what combinations should be used together, and then how forgiving these regimens were. What is the minimum number of doses that you would need to have in order to demonstrate efficacy? Now finally, what about the right time? So for on-demand PrEP, we are really looking for drug concentrations to be at the site before exposure to virus, but there's the potential for drug concentrations to not necessarily achieve maximal effect or maximal concentration before virus exposure. And then how much drug do we need after virus exposure? What is the viral tail, for example? We talk about a PK tail. What is the viral tail in mucosal tissues? Well, we really don't know the answer to that, but we do know in cervical tissue, in a culture system, replication-competent HIV can be there for about a week. So if we make the assumption that we need maybe about a week's worth of coverage after exposure, let's take a look at some of that pharmacokinetic data. So if we're just doing daily dosing of drug in the female, for the female genital tract or for rectal tissue, we see that we get a maximal protection by dose 2 in rectal tissue and maximal protection by dose 3 in the female genital tract. But you can see here, even after a single dose, we see fairly good proportion of the population achieving that target ratio. So we measured, or we modeled the IPRGay dosing strategy for both colorectal tissue and female genital tract tissue, again, looking at the percent of the population that would achieve this target exposure. And in both colorectal tissue and the female genital tract, we found that the majority of the population with this 2 doses before and then a few doses after exposure would achieve an effective concentration. The difference lies in what happens after the fact. So because we have very high concentrations in colorectal tissue and we have lower DATP and DCTP in colorectal tissue, we way overshoot this target exposure. And so we have a good proportion of the population at that target exposure for an extremely long period of time. We don't see that with the female genital tract. So 3 days after the last dose of Truvada, we see already that 20% of the population has lost its target exposure. 5 days, there's about 60% of the population losing its target exposure. And then at 7 days, about 85% of the population has lost its target exposure. So depending on what this viral tail is, we may need some extended dosing here in order to protect female genital tract tissue from infection. So in summary, does the pharmacology support on-demand PrEP? Well, we've looked at the right site, right concentration, and right length of time. And I would say yes, if your drug or metabolite can quickly reach the sites of infection, which we do see with tenofovir and FTC. And if they don't get into mucosal tissue so well, they absolutely need to concentrate in regional lymph nodes. If your compound achieves potent concentrations, we don't exactly know what potent means. We use the IC95. Perhaps we could use an IC50. The clinical correlate of that is still unclear. And that your compound has a long residence time to cover any residual virus. There are some caveats with this. There's still a question of viral trafficking, and can virus be trafficked to sites of low drug concentrations? And are there other physiologic processes that could potentially overwhelm the effect, like inflammation? We've heard a little bit about the microbiome as well. And so with that, these are my acknowledgments and funding sources to those who have helped generate this data, and I thank you for your attention.
Thank you very much, Angela. That was great. Okay, so we're going to be taking questions after all the 4 presentations are done. So if you've got questions, please just note them down. Don't forget them. We're going to deal with them in due course. So our next speaker needs little introduction for those of us involved in the PrEP field, because he did the first study that showed that oral PrEP protects against HIV. So we have in our midst today Professor Bob Grant, who is Professor of Medicine at the University of California. And he's also with the Gladstone Institute, and is also the Chief Medical Officer of the San Francisco AIDS Foundation. In leading the IPREC study, in many ways, he helped all of us in the field understand the real challenges in undertaking the study, but also in how to ensure that you really understand what is going on in the study. And when we learned of the IPREX results, we also understood the complexity of the behaviors involved, we understood the drug levels involved, and we saw the great results that we've applauded. So to Robert and to Bob, who's now just finished a stint at WHO in the PrEP department, we hand over to you. And he's going to talk to us about some of the clinical studies on intermittent PrEP.
Thank you, Slim. And I just have to say this anecdote. Capriso 3 and IPREX presented in the same year, and Science likes to collect a list of the top 10 scientific findings of the past year, and we were both listed there, so I was very proud to be listed with Slim. But none of us were listed first, because that was the year that Homo sapiens DNA was found in Neanderthal genome, and that obviously is a much more important finding than... These are my disclosures. So PrEP usage recommendations are embodied in the approvals that are received for FTC TDF PrEP. And I just wanted to list a few of the available approvals here and what their usage recommendation is. The US FDA and US CDC recommend daily use of PrEP, as does the EMA and Health Canada. Importantly, as I reviewed all of these normative and regulatory agencies only reviewed IPREX and Partners PrEP as their primary file of data that they were reviewing. In the case of Health Canada, it didn't look like they received the IPREGATE data, so they didn't review it. And so, to some extent, the recommendation for daily usage isn't evidence-based or even based on an evidence review. It's based on the file that they got at the time they were asked to approve FTC TDF PrEP. The European AIDS Clinical Society in France do recommend daily PrEP as well, but they both say that on-demand dosing may be used among MSM. And the WHO, in their toolkit just released yesterday or last week, indicates that daily is the current recommendation, but that on-demand PrEP they regard to be safe and effective for MSM, but that it has not been evaluated for others. To be sure, in the WHO meta-analysis now published in AIDS, the IPREGATE data was reviewed. This was a meta-analysis done in early 2015, and so you can see in the red box here that daily versus intermittent dosing was evaluated, and there was a signal toward greater efficacy with intermittent or on-demand dosing, with the efficacy of 86% versus an average efficacy on an intention-to-treat basis of 46% with daily dosing. However, because of the small sample size of IPREGATE, only 400 compared to 17,000 that have been enrolled in daily studies, this interaction did not reach statistical significance, and it remains unclear whether this signal toward greater efficacy with on-demand dosing is important or statistically significant. But I'd like to highlight that this was the data that was available in early 2015, and at this conference and other recent meetings, we're seeing more and more information about on-demand dosing of FTC TDF PrEP in clinical practice. From France, for example, we saw on Sunday a preview of what's going to be presented more fully on Wednesday, which was an experience with 1,581 MSM, which is 57% of the PrEP cohort in France as of the most recent analysis. And among that group, there was no HIV infections among people who had picked up their drug, and that gives us a confidence interval around that zero incidence of 0 to 0.003. The Montreal cohort, clinical cohort now, is also describing 225 people, all MSM. That's 22% of all their PrEP users. And again, zero infections with relatively tight confidence intervals. And so if we combine these, we have good confidence that there's full or near full protection associated with on-demand dosing in now what is a very large number of MSM, 1,806. Now, how many infections might we have expected? Well, that's somewhat speculative. We have to reach to historical controls. But both of these were IPREGATE sites where the incidence of HIV was 6.6%. And at least in the Paris experience, the French experience, 35% of the MSM in clinical practice had had STIs in the last 12 months. And so we think that this is still a very high incidence cohort. And so we might have expected something like 119 infections to occur, and yet we saw none. And so I would argue that this now is a substantial clinical database associated with very high levels of efficacy associated with on-demand dosing of PrEP in MSM. There's more information coming out clinically about who prefers PrEP versus not. I indicated that 22 to 57% of MSM will prefer on-demand dosing when it's offered by a clinician who is experienced and familiar and confident with both modalities. This is data from Montreal showing that MSM in serodiscordant relationships were somewhat less likely to choose on-demand PrEP, but that makes sense. They're sleeping with someone with HIV. The other tendency was that people with fewer casual partners preferred on-demand PrEP, and again, that makes sense. Other information that's coming out is about costs and cost effectiveness, and this again is an analysis based in Canada, which I think represents the cutting edge of this kind of analysis for several reasons. One is that they're deriving costs from actual databases, real data from their public health system and their health system, and they also include direct and indirect costs of treating HIV versus providing PrEP. And what they're showing is that on-demand PrEP is cost-saving in either the least expensive or the most expensive scenarios, substantially cost-saving. Now, the PrEP costs are up front and the HIV treatment costs are lifelong, and so arguably they should be discounted. But even when you discount those future HIV costs at 3% per year, there's still cost savings associated with the expected implementation of on-demand dosing. And that's critical. This is more than cost effectiveness, this is cost saving. So ministers that may be reluctant to allocate the funds can be told that they cannot afford not to provide PrEP in a scenario where it saves them money in the end. Just wanted to remind us that the clinical drug concentration effect analysis for MSM was strongly supportive of the on-demand regimen. This is data from IPREX-OLE. We didn't see any HIV infections in MSM in transgender women who had drug levels commensurate with use of 4 or more tablets per week. Importantly, among people who were using 2 to 3 tablets per week, we saw an 84% reduction in risk. In fact, there was only 1 infection in that range. And even using less than 2 tablets per week, there was a signal towards some benefit, but certainly not enough to make a recommendation. Now, the situation, we think, may be different for women, and this is data from the FemPrep study using pharmacological analysis done by Angela Kashuba's lab. And in this analysis, we see no infections among cisgendered women if drug concentration suggested use of 6 or 7 tablets per week. So there's no infections or seroconverters above that top dotted line. But we did see 9 infections in cisgendered women who had drug levels indicating use of 4 or 5 tablets of FTC TDF per week. This is a type of infection that we simply did not see in MSM. And so I think that this is clinical information that suggests that the relationship between dosing and full protection is different for vaginal exposure than it is for anal exposure, and that higher levels of adherence are needed. There have been some clinical studies looking at FTC TDF non-daily PrEP in women. These are 2 early pioneering studies sponsored by IAVI that were done in Uganda and Kenya, randomized different groups 2 to 1 to receive oral FTC TDF versus placebo, and then re-randomized in a factorial design 1 to 1 to receive a recommendation for daily dosing versus a regimen of twice weekly with a post-intercourse boost. 4 months of follow-up. Sex events were monitored by SMS text messaging daily and by interview, and dosing was monitored by MEMS. So really state-of-the-art monitoring. And what they showed actually was the number of sex acts reported per week were roughly the same between MSM and heterosexual serodiscordant couples. In fact, there was a trend toward more frequent sex among the heterosexuals versus gay men, just FYI. Daily adherence was remarkably high in these African cohorts to PrEP, even in this placebo-controlled trial in both groups. The problem really was with the post-exposure dose, and this was not expected. We expected that to a certain extent sex would not always be planned and that the pre-exposure doses would be the more challenging doses to take, and yet the problem we really saw was in the post-exposure dose. And the reasons given for missing a dose were being away from home, not having pills, forgetting, change in daily routine. So these early studies inspired our HPTN 067 design, which randomized women, including transgender women and MSM, to either a daily regimen of FTC TDF, a time-driven regimen of 1 tablet twice a week with a post-sex boost, or an event-driven regimen of 1 tablet before sex and 1 tablet after sex. These regimens were designed based on animal studies that Walid's group had done and before the IPREGAY results had been published. To be sure, in this study, everyone received open-label PrEP. Dosing was monitored using an electronic device that sends real-time messages to a server called WisePill, and we called people every week for a brief conversation about when sex occurred. The study was independently powered for separate analysis of MSM at Harlem and Bangkok and women in Cape Town. Tim Holtz, who's speaking after me, will describe the MSM experience. I'm focusing on Linda Gale Becker's results from Cape Town among women. Our primary outcome was coverage of sex events with pre- and post-exposure dosing. And these are the results of the primary analysis. And you can see that 75% of sex events in women were covered with pre- and post-exposure dosing when a recommendation was made for daily dosing. And both of the non-daily regimens, there was a step down in coverage from 75% to about 50%. Interestingly, again, as we saw with the IAVI studies, the problem is not with the pre-exposure dose, it's mainly with the post-exposure dose. And the reasons given for missing post-exposure doses were developed by Revea Miko and Linda Gale Becker and their colleagues. And essentially what women said was that the dose was hard to take because they were not at home often. There was concern about disclosing PrEP use to a partner. The pills were not with them. There was a change in routine. Or really the main reason was that they were just not in the mood to worry about HIV. They may be worried about other things, but worrying about HIV in the post-coital period just wasn't happening for a lot of these women. The PBMC measurement of drug levels was really commensurate with the self-reported coverage. So we saw good drug levels in daily and really a substantial follow-up in both of the non-daily arms. So in conclusion, and this is partly my view, on-demand FTC TDF dosing among MSM is ready for regulatory and normative review because I think it is proven to be safe, effective, and cost-saving. The accumulated clinical experience adds confidence and still shows no on-demand bona fide PrEP failure. It's supported by PK modeling and concentration effect analysis, provides an integrated guidance on how to start and stop PrEP around parties, changes in relationships, and drug use. 22 to 57% of MSM prefer on-demand dosing if both are offered by confident clinicians. And the use of the post-exposure dose can be challenging, and I think that's clear, but the on-demand regimen involving 2 post-sex doses really helps. On-demand dosing of FTC-TDF for women, I think, is much earlier stages. Coverage was higher in the daily arms of HPTN 067. We know that women, at least in Africa, are less commonly planning for sex, and the PK analysis has been presented. Thank you.
Thank you, Bob. That was terrific. So now on to our last speaker, Dr. Tim Holtz. He's a medical epidemiologist working for the US CDC. He's based in India at the moment, where he's the director of the Division of Global HIV and TB Office. Previously, he was the program director and chief site investigator with clinical prevention research activities on HIV and STDs with the US CDC at the Emory Clinical Trials Unit, where he was directing clinical trials of PrEP. And also amongst people who inject drugs, as well as men who have sex with men and transgender persons. So, Tim, you're going to tell us about the coverage, adherence, and acceptability of intermittent event-driven oral PrEP. Thank you.
Thank you, Sheena, and good afternoon. I'd like to thank the session organizers for the chance to present this data on behalf of my co-investigators in HPTN 067, Dr. Linda Gale Becker and Dr. Sharon Mannheimer, as well as protocol chair Dr. Bob Grant, who's presented the background and methods of this study. Although currently with the Division of Global HIV/TB India, this work was conducted by the CDC's Division of HIV/AIDS Prevention and the CRS at the Thailand MOPH-CDC collaboration. I have no conflicts of interest to report. Today I will present the coverage, adherence, and PK data from HPTN 067 in MSM and transgendered women from Bangkok and New York City, Harlem, including some qualitative results. One short note on a methodologic difference between the Bangkok and the New York City site. The CDC, CRS, and Bangkok tested PBMCs for tenofovir, and Columbia used dried blood spots, which will not be presented. I will then discuss some studies that have looked at the acceptability of intermittent PrEP and finish with a short discussion of available methods of assessing adherence in intermittent regimens and issues around maximizing adherence. Due to the time constraints today, I will not be presenting the respective site consort diagrams, but they are available upon request. The majority of the 178 TIE participants were in their early 30s, and nearly 90% had completed or were still in college, whereas the age distribution in New York was more widely spread, with an equal number below 24 and over 40. There were 5 transgender women and 2 transgender queer participants, with the rest self-identifying as male. All participants in Bangkok were Thai, and 70% of participants were African American in New York City. Multiple responses on race were allowed there. In Thailand, only 11% were unemployed, whereas in New York City, over 2/3 were unemployed, and participants also included some students. There were no statistically significant differences between the 3 arms for any of the participant demographic characteristics collected. As Bob described, the primary outcomes in this trial were coverage of sex events, number of tablets required and taken, and self-reported side effects. At our Bangkok site, PrEP coverages were similar in the daily and time-driven arm, with no statistically significant difference between these arms. Both coverages were statistically significantly greater than the event-driven arm at 74%. Among people randomized to non-daily dosing in all cohorts, the missed dose required for full coverage was usually the post-sex dose, as Bob has previously discussed. In New York City, PrEP coverage in the daily arm was significantly higher at 66% than the time or event-driven arms, but there was no significant difference between the time and event-driven arms. The New York City site saw nearly double the number of missed post-sex doses than the Bangkok site. The study computed a single adherence number, which is tablets taken over tablets required, over the follow-up period for each individual participant, and used t-tests to compare mean adherence between the arms. In Bangkok, overall adherence, which is noted in bold, which is not the median, was greater in the daily arm compared to the time or event-driven arm, although there was no statistically significant difference between the daily and the time-driven arms in adherence. Compared with daily, the number of doses required for full adherence was reduced by 57% in the time-driven arm and by 80% in the event-driven arm. In New York City, overall adherence was significantly greater in the daily arm compared with both time or event-driven arms. And in both sites, note the considerably lower adherence in the sex or event-driven intermittent dosing arm. Among those Bangkok participants reporting sex in the past 7 days, the proportion of MSM and TG in Bangkok who had detectable drug levels associated with pre- and post-sex dosing was equivalent in all 3 arms. There was no statistically significant difference between any of the arms at either 10 or 30 weeks. And in each arm, at both time periods, we found over 90% of participants had detectable concentration of tenofovir diphosphate, defined as 5.2 femtomoles. This confirms good coverage with these non-daily regimens. In New York City, analysis of drug concentrations in the DBS, as I noted, provided additional evidence that PrEP coverage was lower for participants on non-daily regimens. There were 12 seroconverters in all 3 sites in HPTN 067, but only 4 in the 2 MSM sites. In a recent analysis published by Sive and colleagues, 3 of the 4 seroconverters had received TDF FTC in the once-weekly DOT phase, which was prior to randomization. The other participant infected after randomization had low adherence in the PrEP regimen. 1 participant had TDF FTC resistance. TDF FTC mutations were detected by NGS. Therefore, in HPTN 067, participants who acquired HIV infection had not started PrEP yet or had low or suboptimal adherence, and drug resistance was observed with limited PrEP exposure. We collected qualitative data from 38 Thai participants who joined the ADAPT study in Bangkok. I will not be presenting the data from Harlem. We conducted 6 focus group discussions with 32 participants and 6 key informant interviews. Grounded theory and content analysis were used in this study component, and today I will only have time to review our results for the event-driven arm. Participants in this event-driven arm reported that this regimen worked well if they had infrequent sex events and an ability to plan for sex or having control over planning for sex with their sexual partners. The pro about this regimen was that fewer doses were required. On the other hand, it was difficult for participants to plan for sex, to carry tablets, and to hide them from others. It was confusing to some to count the tablet taking time by the hour, and the regimen was complicated or difficult to follow. So in summary, large proportions of MSM and transgendered women in our study took PrEP consistently when given access in a clinical setting, with coverage and adherence and drug concentrations highest in the daily arm. In New York City, coverage was suboptimal. However, in our Bangkok site, the time-driven dosing arm provided coverage of sexual exposures and adherence that was comparable to daily dosing and spared a considerable number of tablets required. In non-daily PrEP in both sites, incomplete coverage was mostly related to the lack of post-sex dosing. In Bangkok, using the indicator of long-term drug coverage, tenofovir drug detection was high in all three arms at weeks 10 and 30, and in New York City, daily PrEP arm participants had the highest levels of tenofovir in DBS. Our study indicates the feasibility of non-daily regimens in this population with no evidence of risk compensation. So what do we do about the acceptability of intermittent PrEP when framed against daily PrEP as a choice? And two published papers from the last year from PrEP demonstration studies have showed that participants in these demonstration models and consultations that roughly 3/4 of persons choose daily PrEP. Those that choose daily over intermittent in both of these studies shown here were younger, they had either multiple risk factors or had higher proportion of casual partners or were more likely to report serodiscordant partners. Bob has already mentioned the study in Montreal, which has contributed to the evidence base. In Holland, they reported more anal intercourse partners, and in Montreal, less planned sex. And as everyone knows, in the ANS IPRGay study of on-demand PrEP, investigators found a relative risk reduction of 86%, with participants taking a median of 15 study pills per month. They also looked at adherence, which is seen on the following slide here. Published in AIDS Care in 2016, the ANRS team examined the results of an adherence measurement of both on-demand or intermittent PrEP, as well as condom use. And over 70% were adherent to the on-demand PrEP schedule, with the other 29% not interested in PrEP use at all, resulting in over 83% of participants protecting themselves with either PrEP or condoms, and no risk compensation. The ANRS metric of coverage found that scheduled PrEP use was reported by roughly 60% of those reporting PrEP use during their most recent intercourse. The open-label extension cohort study of IPRGay was published, I believe, yesterday, including 361 participants with a median follow-up of 18 months and a median of 18 study pills per month, and they found an HIV relative risk reduction of 97%, highly effective. This was noted by Bob. But there was an apparent increase in anal sex without a condom from 77 to 86%. And also, this team has a poster of the effective rollout of on-demand PrEP in France with over 3,000 participants, and I believe that poster will be Wednesday. So I wanted to finish by touching on the subject of adherence measurement and how best we might assess participants' use of PrEP in any subsequent study of intermittent iPrEP. And this table was published by Haberer in 2015, noting that in adherence measurement there is no gold standard. All of these methods have weaknesses, and the context will determine if more than one measure is needed to understand adherence behavior. Although the easiest to collect, subjective self-report, has not correlated with drug levels in several PrEP trials, as it may overstate adherence. And self-reported adherence can be improved, however, through SMS reminders and surveys of short recall periods and questions that have been validated, as we used in HPTN 067. Pill counts, either clinic-based or unannounced, are inexpensive but labor-intensive and subject to manipulation. Pharmacy refill records provided a more objective approach that has performed well in predicting viral suppression in ART programs and may be feasible for routine clinical work, but are not as applicable in drug trials. Electronic dose monitoring, or EDM, is the only method to assess patterns of use, and they have been shown to be a more accurate measure, including in our HPTN 067 trial using WisePill, although EDM methods require adherence to the device and can present convenience, stigma, and technologic challenges such as we experienced in Bangkok in data that I was not able to present. Drug levels, which we did use, are highly sensitive to detecting use and correlate well with HIV protection. However, these have not been well studied for routine use outside of trials, and they are subject to variation and are expensive. So in the end, multiple measures will be used to help inform adherence, as they can be compared to assess the most informative, accurate, and affordable measures in the less resourced context of implementation. So lastly, how best to maximize adherence in intermittent regimens, and I suspect this may be a topic of intense discussion in the following time period. There are SMS reminders which can monitor both PrEP tablet taking as well as sex events and can incorporate incentives as well. Weekly check-ins, as we used in our trial, can provide support to clients, and this is largely done by phone. And then regular visits with key messaging are also likely critical. So in conclusion, per CDC and the U.S. Public Health Service guidelines, as Bob noted in his comprehensive slide about the normative organizations that have produced guidelines, at least as far as CDC is concerned, daily dosing has been proven effective at reducing the risk of HIV infection in multiple trials and is the FDA-approved regimen. It provides the highest level of protection and the most forgiveness for missed doses and fosters daily habit formation. But as was previously noted, we also recognize that the European AIDS Clinical Society has endorsed daily and non-daily sex event or event-driven on-demand dosing for some populations. Among those with fewer exposures and those who can plan for sex, and I don't know how many of those that includes in this audience, intermittent dosing of PrEP could result in fewer tablets required to achieve comparable coverage. However, one would lose that margin of forgiveness for missed doses, as we found in our 067 study that drug concentrations were lower, data not presented. We have shown that non-daily regimens are feasible and could provide an alternative for those who are able to adhere. I'd like to acknowledge HPTN and my colleagues listed here, as well as dedicate this talk to our former deputy director at the Sealam Community CRS, whose commitment to serving all those affected by HIV continues to inspire our team. Thank you.